Welcome to Neurosalience, the OHBM podcast. Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bandettini. Here I interview brain scientists and discuss their insights and their work, as well as the latest advances in brain mapping. Today, I'm talking with Dr. Lily Mujica-Parodi. She's the director of the Laboratory for Computational Neurodiagnostics, LCNeuro, at Stony Brook University. LCNeuro's research focuses on the application of control systems engineering and dynamical systems to human neuroimaging time series with neurodiagnostic applications to neurological and psychiatric disorders. Dr. Mujica-Parodi received her bachelor's degree in philosophy and physics from Georgetown University in 1992, her Ph.D. in philosophy from Columbia University, and carried out her postdoc in clinical neuroscience, neuroimaging, and psychiatry, also at Columbia University. One of her lab's primary goals is to understand and identify key points of failure in the regulation of neural control circuits, which, depending upon how they break, lead to signs and symptoms that cluster as distinct psychiatric diagnoses. This work has shed light on the prefrontal limbic circuit and how it computes potential threat in the face of incomplete sensory data across a clinical spectrum that ranges from pathological fear to recklessness. Her lab analyzes time series coherence as an indicator of network stability. This stability, she has shown, decreases with age, yet increases with the introduction of an alternate energy source, ketones. And it's directly related to behavior. In addition to stability, network structure also changes with available energy. These insights may shed light on deficits associated with diabetes and aging, and point to ways to alleviate some cognitive symptoms. Her team has built a flat platform called neuroblox for creating and testing network and node interactions using specific transfer functions for nodes to determine how the brain computes. She constrains these models with brain activation data and behavior, and has shown that this is a potentially powerful approach that moves brain mapping into dynamic brain modeling, shedding light into specific mechanisms of psychiatric disorders at the circuit level. In this conversation, we cover all this ground as Lily explains her research. A few things characterize Lily. She's deeply original, thinking consistently from first principles. She's fiercely independent and has a broad grounding in a wide range of skills. And she's also intellectually fearless. I truly enjoyed this conversation and hope you will too. All right, welcome, Lily. Lily Mujica-Parodi. Welcome to the OHBM Neurosalience podcast. And
thanks so much, Peter.
Yeah, well,
pleasure to be here.
Well, it really is a pleasure to have you. And I've been wanting to interview you because I think I've known you almost a decade. I think I met you maybe in 2013 or 14, something like that. And I've always felt that what your work in fMRI and other domains represented was something that was, I always felt it was extremely unique, and it always had and it was unique because, it wasn't just, oh, I'm just doing this or this, you have very specific questions in mind. And it was unique also because of, I felt two things. One, there was an aspect of understanding metabolism, but also an aspect of, you bring to bear your physics background on really careful modeling of what generally people feel is sort of defies modeling, but you've been doing a great job with that. And I think it's worth talking about as far as the way the field could go with that approach. So just to begin, and this podcast will hopefully cover everything from, computational neuroscience, metabolic neuroscience, anxiety, and maybe clinical applications. So just to begin, let's look at a little bit at your history. Your background, I actually was, and correct me if I'm wrong, you received a PhD in philosophy, and so what was your path between that and what you're doing now, as far as and how does philosophy help you in your research right now?
Well, so first I should say, so the type of philosophy I was focusing on was mathematical logic.
Okay.
So it was, in some ways, I would say, kind of closer to math.
Yes.
It's a little bit close to set theory. And I think one aspect of working in a field that's very proof-based, very analytical, is the whole kind of way of systematizing knowledge. So the way I love to work, which, the way that feels most intuitive to me is starting from first principles, and, starting from some set of premises, and some set of inferences, and then deriving sort of intermediate lemmas, and then using those lemmas to then, construct a proof that comes to some conclusion. And that imposes a kind of analytic structure to your thinking. And in many ways, I have really thought about my research in those terms. So if we start from the beginning and we think about what we actually know and what we don't know, people may be surprised to know, what the limits are, the boundaries are of what we really have hard knowledge of, as opposed to, sort of cultural acceptance of,
Yes.
And so, but then in addition, you can ask, what are the intermediary stopping points, the lemmas that we have to establish in order to then get to our end goal? So I would say that is kind of a way of thinking that informs my experiments, and also in thinking about my career trajectory. And that might be a little unusual.
Yeah, and I think it's an advantage, actually. As a matter of fact, actually, I was even listening to one of your talks recently, and I think it was from 2016. At the very end, you say, the field needs more philosophers, or physicists or engineers, but the philosophers part is exactly this sort of thing, where I think that's what you meant in this regard. And I totally agree. I actually think that usually people come into the field very empirical, like they first need to learn a lot of things, and then they start just doing experiments, and they realize there's kind of a question there, and they're trying to do it, and they try to get information, but having this sort of a more formalized structure in your head as to what you're really doing, and how you're asking questions, and what you're assuming and what you're not, I think really would help me at least. So yeah, I think it's helped guide a lot of what you're doing in that regard.
So I would say the other aspect in which it's informed the way I think it is also kind of having a meta appreciation for the field of science and how it operates. And maybe this is sort of an outsider's perspective, but I think rather than sort of jumping in and starting with some training as an undergraduate that then is pursued as a graduate student and then a postdoc and a professor, to me, it's been interesting to see the degree to which the mores of experimental science in the form of hypothesis testing sometimes go a little unexamined, right? Even in terms of dealing with problems like confirmation bias.
Yes.
So I would say those are the sorts of problems that also I think are part of the field, but maybe have been a little underappreciated.
Yeah, and I think we are catching up. I mean, I think that there's sort of a collective appreciation of these sort of errors that can happen, and so I think I like to think that the field is evolving in terms of how we're asking questions and how we're answering them as well. But so so as far as also your training, so were there any, just to cover a little bit more of your background, were there any specific like advisors, or even so I'll lump two questions into one. I mean, were there any advisors or even key moments in your training that you decided, you know, that were very important in terms of deciding to go into brain imaging as far as that's concerned? And were advisors and collaborators helpful?
So I would say that my thesis advisor, Richard Friedberg, he was in the theoretical physics department at Columbia. So I mean, I was in philosophy, but my thesis advisor was in theoretical physics, and and he had a really kind of lovely way of thinking about problems and discussing problems. Very supportive of thinking about other fields as well. When I transitioned to the medical school at Columbia, Donald Klein was an excellent mentor. I don't know if you knew him, but he was really one of the founders of psychopharmacology, and very careful, kind of critical thinker. And I got a lot of very good training and having many discussions with him.
Okay. Oh, okay. All right. Well, all right. So let's go forward to, you know, your research. I think one of our first conversations regarding some of your research was in the whole area of diet and metabolism, and I think you know, you have two recent papers that were really great. But you've been thinking about this for a long time. I mean, two papers in PNAS in 2020 and 2021. I guess we could start with that. Just basically looking at measures of brain synchrony, determining how they're related to brain metabolism, how it changes when you age, but also how it changes with diet. And so, and you have, and the results are just are really clean and really interesting. Would you mind maybe, without, you know, focusing on any one paper or the other, just sort of generally describing your research in this area and what you found? You know, specifically, I was intrigued by, you know, the effects of, you know, ketones and the theory that, and the sort of the really nice sort of construct that a lot of cognitive decline with aging is simply that the brain is less efficient at using glucose, and how that breaks down the networks in specific ways. So yeah, I'm interested in you describing that.
Yeah, I mean, there's a truism in neuroscience that, you know, the brain is 2% of our body weight, but consumes about 20% of our energy, right? And so it's a very energy intensive organ, and the assumption is that we are you know, the brain is constructed in ways topologically that it sort of maximize its efficiency in terms of use of energy. So the question is, so that's very interesting, but then, you know, how do you operationalize that? You know, how do you go beyond that and really start thinking about the mechanisms by which energy utilization actually changes functioning? And so the only way to really think about this is as a sort of a multi-scale model, where you have this bottom layer at the neuronal scale that then sort of propagates up to the whole brain scale. And one of the things we've been very interested in is constructing those sorts of models, these generative models that can show how small changes at the micro level have huge emergent effects at the macro level. So one of the things that we know is that there is this hypometabolism that occurs as we age. It's true not only for people who have clinical symptoms of dementia, but you start to see beginnings of this even in people who have mild cognitive impairment. And so the thought is that this might be sort of a progressive issue, that it's not that all of a sudden you wake up and you have dementia, but there might be kind of a long degenerative process that occurs over time. Certainly, we know that in terms of peripheral insulin resistance, so you have your glucose insulin regulation, and it's a control circuit. And anytime you overtax a control circuit over long periods of time, over time that control circuit starts to become lax, it starts to break down, and the regulation starts to become less tight. And so generally, we know that if any person follows a high glycemic index diet for a long period of time, eventually, to some degree, they'll become insulin resistant. Whether that happens faster or slower has a very strong genetic component. Some people are naturally more sensitive than others, but it's a general feature. And so if we think then about what's happening to neurons, so there are many mechanisms by which the brain might run out of fuel. So the fact that it's hypometabolic doesn't imply necessarily insulin resistance. It's one of several potential mechanisms. But the insulin resistance seemed like a very obvious candidate to be looking at. Even if it turned out to be wrong, at least then what you do is you'd exclude it and look at other potential mechanisms. Yes.
Oh, one thought that occurred to me too, though, with diabetes, for instance, if you have insulin resistance, but you also have potentially vascular effects as well.
Right. Exactly.
Potentially. So.
Yeah. So I will get to that. I mean, we've definitely been considering that as well, and certainly in the context of fMRI, the vascular effects are very relevant. So what we decided to do was to start looking at the impact of insulin resistance on neurons at the microscopic scale, and also look at what's happening at the macroscopic scale with fMRI. We started by looking at healthy individuals across the lifespan. And we found that there was this destabilization of networks, where destabilization is really a very straightforward thing. We're looking at the correlation matrix, the adjacency matrix at any particular time, and then asking how is that changing over time. And in general, when you talk about stability or instability of networks, it makes it sound like one is good, the other is bad. And of course, that's not the case. I mean, a truly stable network would be one that was dead, but a truly unstable one would also be a problem. And so there's sort of an optimal, U-shaped curve. But what we find is that in order for there to be processing within some particular circuit, the network needs to be activated long enough for that processing. And interestingly enough, we found that as people age, those networks do become destabilized. And we were able to replicate this in several large-scale data sets. So initially, we were a little bit surprised that it was not just that it became destabilized with age, but it really had this very characteristic sigmoidal shape to it. So we initially showed this in the CAMCAN data set, which is 636 subjects, and then later replicated it with HCP, which is another 725. And then also with the Leipzig, which is another 292. And in general, you see this behavior. And certainly, when you see this sigmoid, you'd like to know, well, what's driving it? So the base of the sigmoid starts in the late 40s. And then you see this very steep destabilization. And if you look at the very end point, there's a precipitous drop. And then it becomes hyper stabilized at the very end. And what we find is that this sigmoid, which seems to be replicating over and over again in healthy individuals, when you look at people with APO4 positive status, the sigmoid becomes dramatically accentuated. So the same sigmoid, it exists, it looks the same, but it's much more dramatic. So
just to reiterate, as you age, so it's a sigmoid that just kind of goes up. So a measure of network stability, and so it's basically becoming less synchronized
Let-
or network
yeah.
synchronization. So
Yeah.
your brain, and then just to quickly address some things that might be on people's minds, controlling for things like motion and CSF or physiologic noise and things like that. That's usually pretty constant, or might even go the other way potentially. So those aren't factors. It's really is a measure of. So you used fMRI to look at this, and you also looked
originally fMRI, and then we started also looking. There was an MEG data set that we looked at. We've been looking also at EEG, and we see the same behavior. So that suggests, given that it's present also in MEG and EEG, it's present. It seems to be not only vascular. So it might have a vascular component, but certainly the sigmoid is not just driven by the vascular changes. And then the interesting thing is that, so the question is, what's driving this? The thought is that what's happening during the sigmoid is that there is, at the bottom of the sigmoid, basically there's this homeostatic regulation that is happening as the brain starts to run out of fuel. But at a certain point, it looks like there's some degree of metabolic stress, meaning the brain is starting to run out of energy to some degree. The brain is still able to compensate for it, but there is some element of struggle that is happening during, the middle part of the sigmoid. And what we think is happening at the end of the sigmoid is that you hit a breaking point, and at that point, the neurons have run out of fuel to such a degree that they can't survive anymore. And then you see this precipitous drop, which seems to be associated with atrophy. So this is a really important feature, because it suggests that if you are going to treat metabolic stress, there's a window of opportunity for doing it. And it might be pre-symptomatic. Yeah. So none of us thinks that, by our late 40s, we're showing symptoms of cognitive decline, but at the same time, it suggests that if we're going to treat this metabolic stress, we might have to not rely on symptoms as the trigger for us to do something about it.
Okay. Yeah, no, that's interesting. So and the idea then, once again, is basically the regions of the brain that are typically easily able to communicate to synthesize information are, that's sort of that the network fidelity is sort of going down to some degree, and then they're basically trying other connections and strategies, and so that results in a decircuitization to some degree.
So yeah, one hypothesis is that basically the brain is kind of re-optimizing. It's trying to find an optimization strategy that will allow it to conserve fuel by finding cheaper routes. Yeah. That's a hypothesis, and I don't think we have an answer to that yet, but we're starting to see some clues that something like this might be happening. At the bottom scale, I can say, although our results are still preliminary, what we see is that when we're collaborating with Nathan Smith at University of Rochester, who's been doing experiments with field recordings, patch clamp, he'll be doing calcium imaging, and then up to EEG. And what we see at this point with the field recordings and the beginnings of the patch clamp recordings is that there seems to be really profound changes to firing dynamics at the single neuron scale. But then, when you look at field recordings, you see changes in axon conductance velocity. Yeah. And all of those are going to have implications at the top scale for how those networks are formed and then held online. Yeah. So in terms of trying to figure out what the mechanism was, we started using beta hydroxybutyrate because there are lots of potential mechanisms, but what BHB allowed us to do was to isolate the mechanism specifically to GLUT4. Yeah. Right. So if what we're thinking is that there's insulin resistance, the idea is that GLUT4 is not, the signaling with insulin is not working properly, and the glucose is not able to get into the cell. But of course, there's a back door, and beta hydroxybutyrate is able to enter the cell without GLU4. So we started off by looking at healthy individuals who were young. And what we found is that actually, we started off actually looking at dietary changes. So initially we had individuals under fasting state, normal glycemic index diet, standard American diet, really.
Yep.
And then with a ketogenic diet, but just for 1 week. And what we found actually was that even after these really short-term dietary interventions, we see dramatic changes in the stability of these networks. Okay. And then this was replicated with beta hydroxybutyrate for a couple reasons. One is that when you change someone's diet, you're changing lots of variables. And so what we were able to do with the BHB is to control for the dose. It was weight dosed, it was calorically matched. And so we knew that whatever changes we were getting in the network stabilization or destabilization, that they weren't caused by caloric changes. They were specifically related to the fuel substrate.
Yeah. And so what's really interesting about this as well is that ketogenic diets, beta hydroxybutyrate has been used, to reduce epilepsy. It's one of the most effective dietary interventions for reducing epilepsy. So it seems like there could be some relationship there in that regard. So, yeah.
Well, I would say one of the reasons it's a really interesting field to look into is that there's some very fundamental questions we don't know the answers to.
Yeah.
So as for why it works with epilepsy, there's some hypotheses in terms of how it affects GABA, but I think there's still a lot of foundational work to be done.
So they may or may not be related, but either way, yeah. But overall, the hypothesis at least is that this backdoor, this energy source, somehow just amps up or increases the metabolism of the neurons such that the network integrity is better preserved in that regard.
So interestingly enough, the thought is that that the BHB is a better fuel source in some ways. It's a more metabolically efficient fuel source than glucose. What we find is that even in people who don't have insulin resistance, it's stabilizing networks as compared to glucose. That was surprising because it's always been, I think, assumed that glucose is the brain's natural substrate, and how could you do better than glucose? But it does appear to be the case. And when we look again at the field recordings, we see that the conduction velocity with beta hydroxybutyrate, the same stuff that we're giving to the humans, increases above glucose in these neurons.
Huh.
So it seems to be true at all scales. And for me, that was a very shocking result, because we're used to thinking of, well, in a deficit state, maybe this is correcting for something that's wrong, but it suggests that even in a non-deficit state, there may be advantages to utilizing ketones. Interestingly enough, getting back to the sigmoid, what we're finding is that, so the ketones have this very dramatic effect. It's really 30 minutes after they take it that you start seeing effects, and it persists for at least an hour afterwards. At least that's when we stop measuring and we still see an effect. But interestingly enough, so the biggest effect is during the meat of that sigmoid that I was just describing, the period that we think is related to metabolic stress.
Yeah.
But once people get to that peak of the sigmoid, the ketones don't do anything anymore.
Ah.
And so it suggests that, again, there seems to be sort of a window of opportunity for refeeding these neurons, but if you miss that window, it may not be possible to treat it in this way.
Okay. And other interventions, aside from ketones, one might think, oh, well, why not just find other ways of increasing the metabolic rates? Sure, glucose is available, and maybe if you just simply, drinking more coffee or exercise or other things like, people have thought infrared light, helps. What do you think of that? I guess without the fuel source, without effectively using the fuel source, trying to do that seems like it's less effective, but yeah.
Certainly. So exercise is thought to increase insulin sensitivity regardless. So you'd think that anything that would increase insulin sensitivity is going to help address this problem. It can prevent insulin resistance to begin with because it'll blunt the glycemic index of whatever it is you're consuming. Exercise has other effects on the brain in terms of BDNF and so forth. Yeah. So, I mean, all of these interventions have a lot of different pathways. So it's, you have to be careful about isolating those pathways.
Right. But it's really remarkable. I mean, even looking at the network stability and looking at even behavior, like you showed with, giving ketones with typically healthy people, they can, their sustained attention or reaction times or things like that you've measured were, improved. So that was interesting. Yeah.
And then actually, you mentioned this whole issue of the Ising model and this other paper. And again, this gets at the fact that there is a sort of a discontinuous process where, the Ising model is designed specifically to look at phase transitions. And you might ask, well, what kind of phase transition is going on? Interestingly enough, again, the phase transition that is picked out by the model is not occurring in old age. The phase transition was picked out again in the late 40s. So something is changing at that point that is kind of dramatic.
Huh. That's interesting. So right, the Ising, and that sort of brings to mind, the Ising model is the way that you address your fMRI data and perhaps even the other measures, but specifically I saw it in the context of fMRI where it's a very computationally, a lot of people are trying to do this, including myself, trying to find ways of coming up with really good measures of connectivity over time. And windowed correlation is one way, but this model seems like it's elegant and computationally efficient, and it's really sensitive. So maybe it might be something more people could use as far as that's concerned. And I'm kind of, just from a methodological standpoint, it'd be interesting to see if you see the same results with other, the more standard approaches. Maybe not. I don't know. So do you think that? This is another question before we get on to another topic, but that there are parts of the brain that might be, there's certain hubs of communication. There's certain maybe areas where things are controlled, to the extent that you have a reduction in metabolism, there's a chance that some areas might be either influence more other areas, or might have a different metabolic profile in that regard.
Well, with the Ising model paper, what we were seeing is that, there are periods. So if you are to look at your time series over time, at every particular time point, there are periods of high synchronization or low synchronization. And what you find is that during the periods of low synchronization, the networks that get activated are primarily the segregated networks. These are the local networks that are, computing locally. And during the periods of high synchronization, the networks that are added to that are the integrated ones, the ones that are connecting those local networks. Yes. And so what seems to be happening is that during this transition period, there is a kind of an abrupt change between having equal access to both of those functions, which after all, it's not like one is better than the other. They have to operate interchangeably in order for the circuits to work properly. But at a certain point, there seems to be then a bias towards the segregated at the expense of the integrated. And again, getting back to this axon conduction velocity result, if in fact there is a slowing down of conduction along long axons, obviously it's those longer connections that are going to see an effect more so than the short ones. And so this might actually provide a mechanism for that.
Yeah, and I'm glad you brought that up. I mean, I like the idea, the model in terms of, integration versus segregation. And, the long range connections are metabolically more demanding and maybe more fragile in that regard, as far as metabolically. And so, and what are the implications when those long range connections break down maybe before the short range connections? And so that's
that's really the next step, because as you know, most of the work we had been doing previous to that, to getting into metabolism, was thinking about circuits. And we're doing this kind of foray into networks to think about metabolism in the context of affecting networks, but ultimately the next step is to go back to circuits and ask, how is metabolism then affecting the dynamics of these circuits and how they regulate and process information?
Yeah. So that's a perfect segue into the other aspect of what you're doing, and that is, you're doing working in several areas, but one is computational psychiatry. And what I found really unique about your perspective and your work is that you really have been trying to apply rigorous generative models to what you're doing, as opposed to just mapping or looking for biomarkers or things like that. You have very specific hypothesis, very circuit level hypothesis that you're testing. And just before I get into that, I thought, you had a review article in Journal of Neuropsychopharmacology in 2020 that really set things up, I think, kind of nicely, differently than that I've seen other setups. But you basically say, well, there's three types of models. There's sort of a bottom-up, sort of reductionist sort of approach, where you start with neurons and you just sort of build. And the other one is looking at the second one is looking at sort of this emergent properties of any sort of measurable effect, and you're trying to sort of then iterate between building hypotheses that explain that effect. And the third one is sort of interesting, where you say it's more of an information theoretic approach, where you assume that sort of the goal is to optimize efficiency, and how does the brain do that sort of pretty well. And I like that construct, and sort of you're like in the middle, the second one in some regard, as far as that's concerned.
So I think that a lot of this approach was very influenced by the fact that I have a lot of experience modeling systems outside the brain. So I'm very interested in physiological control systems. I teach a graduate course on modeling physiological control circuits. And that would include autonomic control circuits, endocrine control circuits, immune control circuits, and so forth. And what you start to see is that nature doesn't reinvent the wheel every single time it needs to maintain homeostasis. I mean, there's some certain types of circuit architectures that get used over and over and over again in different systems, but also at different scales. And so what you really wind up with is a system of nested control circuits. And I think that sometimes this is insufficiently appreciated, because control circuits behave in ways that aren't necessarily amenable to statistical analyses. So when you look, for example, at a healthy person and a type 1 diabetic or type 2 diabetic, and if you're taking a invasive glucometer and looking at a continuous measure of their glucose over time, what you'll find is that the distinction between the patient and the control is not the value of the glucose. Someone who has diabetes might have a glucose level that is higher than that of a control. It could be lower than that of a control. It could be the same as that of a control. What distinguishes someone with diabetes versus not is the dynamics of the regulatory behavior, not the value per se, or even the average value per se. And so the problem is that statistical analyses are usually looking at some value and comparing that value between populations.
Yep.
But if what is relevant is not that value, but rather the dynamics, it's going to not be very sensitive in identifying the mechanism.
Yes.
So one of the things that that was interesting to me when I first started moving into psychiatry and psychiatric neuroscience is that, people often, when they started doing neuroimaging in patients, all sorts of different types of patients, what was really amazing was that the same areas of the brain were implicated in almost every psychiatric disorder. And what that seems to suggest is that it's not a kind of lesion model where, something gets knocked out, and it's in this population, but not in that population. Depending on where it gets knocked out, then that'll give you different types of psychiatric disorders.
Yep.
It's probably closer to thinking about diabetes, where you have disorders of the same circuit. You type 1 and type 2 diabetes are both dysregulations. They're different types of dysregulations of the same control circuit, where a feed-forward problem gives you one type of disorder, the feedback problem gives you a different type of disorder. But what's really interesting is that if you were to look at type 1 and type 2 diabetics, what you would notice is that they don't seem to have very many features in common. So, type 2 diabetics tend to be overweight, type 1 diabetics tend to be underweight, And what's also very interesting about diabetes is that you see vision problems, you see foot problems, you see all sorts of problems that don't seem to have anything to do with the mechanism that's actually driving the disorder. And to me, that's actually very instructive. It suggests that probably those sorts of considerations are also need to be taken into account in psychiatric disorders, where the symptoms may radically change as a function of the type of dysregulation. And rather than saying, oh, it's this network with this type of disorder and this network with that type of disorder, it might very well be the same circuit that's dysregulated in 3 or 4 or 5 different ways. And trying to understand what those dysregulations are, what those candidate mechanisms are, is ultimately what might be most successful in distinguishing the disorders.
Yeah. Go on. Sorry. Go ahead.
Mm-hmm.
Well, I was going to mention that you have several ways of actually, it was really interesting to see along those lines of probing dysregulation, showing examples of how the brain doesn't come back to its regular state after a perturbation. I don't know if you were going into talking about that or not, but.
Well, I think actually that's also kind of important because there's been such a move towards resting state studies, and it's a little bit of a cautionary note because when you want to look, I said, if you look at a diabetic might look exactly like a healthy control, unless you actually perturb the system and you give them 75 grams of glucose, right, with a glucose tolerance test. And so, although resting state has a lot of potential, it's worth considering that unless you perturb the system, you might not be looking at regulation as efficiently as you would if you were actually upsetting it somehow and then looking at how it returns to baseline.
Yeah. And so how have you been perturbing the system in this regard? You had a couple of really nice papers on basically invoking the ventral medial prefrontal cortex as sort of like this regulator of anxiety and taking information and then giving and sort of speaking to the amygdala. I don't know if you want to mention that in some sense.
When we started looking at the prefrontal limbic circuit, the standard ways of activating emotional responses was to show angry faces and fearful faces. Certainly we started there and then kind of more influenced by the animal research, like Joe Ledoux's work, we started looking at fear conditioning. And I think our most unusual way of activating the circuit was a brief foray into alarm pheromones, where, the problem that I was having with all of our stimuli is that humans are very good at cognitive blocking. If you see that somebody is presenting a stimulus, you understand that that you're being manipulated in some sense. And so I was curious to see if we could activate this prefrontal limbic circuit by unconscious means. And so I was kind of very influenced by some of the animal research on alarm pheromones. We decided we could replicate that, which we did.
Yeah. And so that was a fun study where you basically collected sweat from people who were either exercising or jumping out of planes, presenting it to people and actually showing that there's amygdala activation with this odorless, only the stress-induced sweat. And have there been follow-ups on that? Is there an idea of what the mechanism is? When I was reading those papers, I was thinking, oh, this is, you think that people could synthesize this, or either because it actually didn't invoke, this overt behavior, just like the studies on mice. It's like, with mice, if there was fear, they actually became, manifestly fearful, but here it just simply upregulated.
I'm not sure that's the case.
Okay.
So what happens with the rodents is that the alarm pheromone, it's not, so if you think about, a fear response, let's say the environment in which alarm pheromones evolved is, you have some, mouse out in a field, and one of them detects a predator. Freaking out is not a very adaptive response because it's just going to gain the attention of a predator. So there has to be some way to sort of communicate with others of conspecifics, others of the same species. But what the pheromone actually does, if you look carefully at the behavioral responses, it actually increases, it causes freezing, tracking, sniffing. That's information gathering.
Yeah.
And what it really seems to do is to sharpen perception of threat, but also distinguishing between threat and non-threat. So what happens is that normally we're kind of sloppy about paying attention to our environment, and the alarm pheromone actually seems to sharpen our perception of threat. And we saw something very similar in humans as well. So it didn't induce fear, but I would say it didn't induce fear in the animals either.
Okay. And it's always, I think, one of the really cool concepts that's, everyone kind of knows, but it's worth restating. It's all about, it seems that it's all about homeostasis. It's all about, you gather information, and if you make an error in terms of thinking it's a threat when it's not, you make an error that could affect your survival. If you make the error the other way, you could also affect your survival. And the whole goal is homeostasis. even some people argue that, the manifestation of our own conscious experience is a form of homeostasis in this regard. So it's a really interesting construct for thinking about.
It's actually a very elegant mechanism because it's kind of the processing of the stimuli is a sort of a just-in-time type mechanism. So there's a default excitatory response, but there's this this processing of sensory stimuli that runs in the background. And as soon as the brain has enough information, it then, ideally then inhibits the amygdala. Yep. But what happens, we found in the individuals with greater anxiety, trait anxiety, and then even more in a more extreme way with clinical anxiety, is that they get trapped in this sort of information processing loop. And therefore, then there's this delay in the inhibition of the amygdala. So it really was more of a sensory or information processing problem rather than an emotional problem per se of these limbic regions, which is kind of counterintuitive again, unless you're thinking about it as a circuit as opposed to simply regions that, well, this goes up and this goes down. It's more complicated than that.
Yeah. And are there ways of, so let's say we have, so I think that's the way we're trying to go with brain imaging. Psychiatry is, by all measures, it's relies on just purely behavioral assessment and, really trying to go to the brain. We start by saying circuits. But how do you actually, like in humans, it's all this sort of, subcortical, prefrontal, limbic sort of circuit. How do you further probe this? Do you have to come up with really good paradigms? And then obviously you have to make your generative models, and it's all very nonlinear as well. But how do you actually, fMRI, some would argue that might be not specific enough or just fast enough. EEG, there's all these limitations. How would you further develop this approach? As far as that, maybe you're thinking of that just now.
Well, definitely. I'm definitely thinking about it. The first thing to say is, I think you'd agree, every single modality has its strengths and weaknesses. And at the end of the day, I mean, what you need to do is to use a variety of different modalities and see how they combine to tell you a coherent picture. And what I mean by that is that, so fMRI has, of course, the limitation that it's an indirect measure that's hemodynamic, so you're not measuring a neuronal response directly. But where it shines is that you have access to the entire brain. So it really functions well as an exploratory modality where you have access to potentially the entire circuit. One of the aspects is getting back to my, starting from first principles that has really informed my way of approaching problems is I have a very strong mistrust of assuming that I think I know what the circuit is or what the network is without actually letting the data tell us.
Yeah.
I think that there has been a lot of, let's say, local minima in terms of groups saying, oh, I did an experiment, I found this, and then everybody else sort of looks for the same thing and they find it, but not really considering alternative hypotheses. So, and certainly this happened with the prefrontal limbic circuit, even with Joe Ledoux's work with mice and rats, because the problem was, and this is getting to the limitations of electrophysiology, you have to know where to put the electrodes, right? And so to a large extent, the hypotheses you're going to be able to test are going to be driven by where you're measuring. And since you can't measure everywhere, and because these experiments are hard, you might miss something crucial. And so this whole ventral stream component of anxiety was missed by that early work on the prefrontal limbic circuit, simply because he didn't think to put the electrodes there. And why would he have? So this is where fMRI has something to offer, I would say, and why it shouldn't be ignored. It has to be part of the toolkit. But once you have the circuit, then of course you want the dynamics to come from more targeted measurements. So the way we have been approaching this problem, initially, we know that there are certain dynamic signatures, so let's say entropic signatures of negative feedback loops. So we first apply those, these entropic measures to all the time series in the brain using fMRI. And then identify the voxels that show these very different dynamics. Those then identify what we think are the nodes that matter. And then we use, so I would describe that as sort of a global approach, completely data driven, without any sort of bias about what is important and what is not important, but it identifies what's involved in a feedback loop. And mind you, again, getting back to these statistical tests will not tell you anything about feedback loops. That's not what they're really designed to look for. And then once you've identified the components of the negative feedback loop, then you identify sort of the connectivity in terms of it has to be directed connections. What are the inputs and outputs to each of those nodes? And then from those inputs and outputs, you can derive transfer functions. And what you wind up with is a system of coupled differential equations. And that is your generative model. At that point, you can ask, okay, what if I give a new set of inputs? What sort of behavior ought I to expect? And that's how you start testing the model. And refining it over time. Now, the prefrontal limbic circuit that we had worked on, it took about 10 years we had 226 subjects at all different levels of anxiety, from, as we said, anxious to reckless, everything from clinical anxiety to trait anxiety to sort of healthy, normal people to sort of trait recklessness, and then extreme recklessness. And of course, we had people jumping out of planes, so we had this population that allowed us to test how they responded to this actually dangerous environment. And we saw this dysregulation that occurred that we could sort of track over the entire spectrum. So that suggests a sort of an axis of dysregulation, where different types of dysregulation were associated with each end of this spectrum. And our approach is then to say, okay, this is one axis. Can we consider what are the other axes that are relevant? Where those axes are going to be associated with different basic functions that are psychiatrically relevant. So talking about RDoC, I could imagine an RDoC that's based on some sort of parameter space where you're thinking about these dysregulations for these different circuits is occurring, again, within some space where you could track them as being, like this disorder is in this place in space, and this disorder is in this place in space.
Yeah.
Where the space is defined by the circuits and their regulatory properties.
Yeah, and it might settle into, right. I mean, you might have, 5 different categories of disorders, and they might be 1 or 2 circuits with specific disorders in the circuits. I mean, that would be the goal. I can imagine that happening in some regard. So,
then we decided, all right, well, look, that took 10 years, and we can't spend another 10 years for every circuit, right? Every iteration. So we were thinking about, well, how can we speed up this process by taking what we did and automating it, doing it better? And so in the last few years, we've really been focused on working with computer scientists in thinking about algorithmically, how do we take what scientists do and do it better? And what I mean by better is, in part, do it in a way that's less biased. And here's what I mean by that. So if we think about, 2 extremes. So we can think about a pure machine learning approach, where basically we have a whole bunch of data, and the output is we classify into this type of disorder versus that type of disorder. Well, that might do a great job of classifying, but it doesn't necessarily tell you much about the mechanism. So it is completely unbiased, but in some sense, it doesn't advance the field, really.
Yeah. Yes.
The other extreme is, we have a very clear hypothesis, and we test that particular hypothesis. But the danger of that is that you're guessing. Well, okay, I'm testing that hypothesis, but what about all the millions of hypotheses I'm not testing? And by the way, how many competing hypotheses would have given exactly the same answer, right?
Yeah,
exactly the same dynamics or whatnot. And so you never know if, well, okay, maybe that hypothesis leads to that outcome, but there are lots of competing hypotheses that would lead to the same outcome.
Yes.
So in order to do that, you really need to consider lots of competing hypotheses. And one way to do that is to think about somewhere between machine learning, which makes no assumptions about what the structure is, and complete domination of those structures. You might say, okay, what are the primitives? What are the computational primitives? So if I look at your face and I were doing machine learning, I would classify your face by its pixels, right? But as a human being, I look at your face and I see eyes and a nose and a mouth and hair. And if I want to talk about you, those are the constructs I would use. Yeah. So then the question is, what are the elements that we need to give machine learning so that it can look at all the possible permutations, all the combinatorial possibilities, and test all of these different hypotheses and make predictions, which then allow us to say, okay, these are the candidate hypotheses that are consistent with our data.
Yeah.
And these are the candidate hypotheses that are not consistent with our data. So what it won't do is a one-to-one matching, because there's more than one candidate hypothesis that will be consistent with your data. But what it can do is narrow the search space in an unbiased way, and then allow you to then go and distinguish between those competing hypotheses, where again, the simulations can allow you to sort of optimize over where the differences are likely to be most extreme. Yeah, so and that's really the direction we're working in now.
That is cool. That's cool. I mean, that's really clever. I think that, right. It's sort of an efficient way of searching the possibility space such that it gives you some options that progress you not quite there completely, but to a point where you can then do experiments and test these things. That's cool. And you have, and it's called Brain Blocks, I guess. This is the platform.
Neuroblox.
Neuroblox. I'm sorry. Neuroblox is the platform for studying that. And is that open source, or is that? I mean, you've bin developing this sort of thing. Okay. Yeah.
Yeah, so we haven't released it yet, but yes, it will be open source. And I can tell you something about it. So it was kind of inspired partially by the painful process of putting together models and thinking about how can we make this more efficient. Also, how do we communicate with other people's models? So when we're trying to put together these models, so let's say we want a prefrontal limbic ventral stream circuit or a CBGTC D1, D2 circuit. We have to go to all of these different papers and try to extract the equations and whatnot. And so the question is, can we develop sort of a common grammar in much the same way that these software packages in neuroimaging allowed us to talk about results from different groups because there were common pipelines? Can we do something similar for computational modeling? Where we create these libraries of models and people can then work with what other people have already done and test different elements of them and change something and see how do the simulations change if we test this versus that, what models are replicating over time, in which case they sort of go up the leaderboard, which models are getting disconfirmed by experiments, in which case they go down the leaderboard. And in that way create sort of a crowdsourced consensus as to which models seem to be working and which ones are not.
Yeah.
But again, you can't have many groups working with the same models unless they're speaking a common language. So Neuroblox is designed to be modular. And I'll explain what that means in just a moment. It's explicitly multi-scale, multimodal, and multi-system. So what I mean by multi-scale is it has everything from the neuronal all the way up to fMRI. It's multimodal because it has fMRI, MEG, EEG, local field potential, and so forth. And it's multi-system because one of the aspects that I think is really underappreciated in the context of psychiatric disorders is the degree to which the neural circuits are interacting with other physiological circuits. And there's feedback between the rest of the body and the brain, and that becomes relevant in some cases.
So could you expand on that just a little bit? I mean, that one point, I'm trying to picture like what would be an example of interacting with the body in some sense in terms of constraining the model.
Well, so I mean, so one example that I speaks to me personally because I had Graves' disease. I mean, when we met, actually, I was very symptomatic. So Graves' disease is an autoimmune disorder that affects the thyroid, right? And one of the consequences of this is that your metabolism goes higher. So you're really hypermetabolic and you have very high heart rate and whatever. But I didn't realize this. What I did know was that I was suffering from massive anxiety. And so, if you were to look at me at that time, you'd say, oh, this is a person who's extremely anxious, having panic, attacks and whatnot. You might take me to a psychiatrist and have me talk about my childhood and my conflicts and so forth. But the reality is it was an autoimmune disorder. So it wasn't even about the thyroid. It was really the immune system. Yeah. So, it's really, again, how many psychiatric disorders implicate other systems where we're not even clear to what degree they are driven by the brain or if the brain is simply where that disorder manifests itself in terms of behavior. Yeah. So one of the disorders I'm very interested in is bipolar disorder because it's very much a dynamical, a disease. Yeah. Where you have this trajectory of oscillations or periodicity, and but there's so many disruptions in that disorder. I mean, there are circadian disruptions, right, and there are autonomic disruptions, and it's not even clear whether there's some element from some other part of the body that's influencing the brain, or whether the brain is influencing those other systems. So one of the nice things about the modeling capabilities of Neuroblox is that it allows you to test those hypotheses in silico, and then to start being able to determine what would be the ideal experiment to establish what are the driving mechanisms.
Okay. So let me just ask a question along those lines then. So you have the Neuroblox, and you have some data and that constrains it, and you finally find a good circuit or a good hypothetical sort of generative model that sort of explains it. And then, okay, so now let's say you can use that to, you have then this disparate data like either fMRI or EEG to help kind of diagnose a patient. I mean, maybe that can be used to sort of constrain what's going on, but then the question is, how do you intervene? I mean, maybe the model itself could suggest specific ways of intervening. And then
So the model is also specifically designed. These models have I mean, neurotransmitters are part of the model. So because most of our pharmacological interventions are targeting neurotransmitters, it's important to be able to understand that certain connections, certain components of the circuit are dopaminergic at D2 versus D1 versus acetylcholine and so forth. And so that adds a layer of complexity that I think also will be important in understanding the dynamics. I should also mention that neuroblox is not just coming out of my lab. It's a collaboration with Alan Edelman and Earl Miller at MIT and Rick Ranger at Dartmouth and Hellman at Stony Brook. So the idea behind the neuroblox and the modularity, as I was mentioning, is that one of the cool aspects is that you first have to ask, what are those computational primitives in the brain? So we also in neuroimaging often talk about processing, but in a sort of metaphorical sense.
Yeah.
Because even within, let's say, models like DCM, you have, something is affecting this and this is affecting that, but it's not clear what is the actual processing that is happening. So at some point, if we think that there's actual processing, we have to be a little bit more specific about what's going on. So, in the context of an electrical circuit, if you wanted to look at a circuit diagram in EE, you'd have resistors and capacitors and, batteries and all of these elements that would allow you to construct circuits from them. And so the question is, what are those in the brain? And so that's one of the kind of interesting scientific contributions from neuroblox, is we have to sort of go back and say, even at the neuronal level, how does this processing occur? So in other words, if in our model we want a filter, because we think that this region of the brain is acting as a filter, biophysically, what is the mechanism by which that filtering happens? Or if there's a gain over here, what is the mechanism by which that happens? Right. So we're being kind of careful about that. There's a tendency when you're doing modeling to just make it work. But for us, it's not enough to make it work. It has to also be biomimetic. It needs to say something biophysically realistic about how the neuronal processing is thought to happen.
So right, you can break it down to some degree to, like you said, either gain in terms of, or inhibition, excitation, and so it's interesting to try to figure out, with the concept of calculation, whether that can be broken down into what we know neurons do. I mean, they increase excitability, they decrease excitability, they might.
Well, they do more than that.
Yeah, they do much more than that. But it'd be interesting.
They do more than increase and decrease.
Right.
Is what I'm saying.
Yeah, and it would be interesting to try to make a library of these elements of what they do in that regard to help constrain what it means.
So those are the blocks.
Yeah.
So we have libraries of these computational primitives, which are the blocks. These are coming from, again, the animal literature. Rick Ranger has taken the lead in, this is what he does. He's been doing it his whole career, really identifying these neuronally generated processing units. And those then create the lowest foundation here of these circuit diagrams, where multiple components of these blocks then can create, super blocks.
Yeah.
And then multiple of those super blocks then also combine. And so again, you have the capability for nested control circuits where you can go from the lowest level all the way to the highest, most emergent scale. Yeah. But at every scale, you're talking about a control circuit. And the reason why you want that ability to go from the lowest scale to the highest scale is getting, again, back to metabolism. Yeah. Metabolism is, the mechanism for metabolism is happening at the lowest scale, right? Mm. So if we're going to be changing parameters, they're going to be at the very bottom layer. Yes. So any model that's going to be something you can adjust has to be adjustable at the lowest level to be able to ask, what happens if we change metabolism here? What is going to be the impact for the circuit behavior, the regulation of the circuit as a whole?
Yeah. That's cool. I mean, right, the emergent behavior of metabolism change in some sense. You could predict this and have testable models for what would happen. So that's really a nice. It seems like it's kind of like it's a lever for making more of an impact in terms of understanding this huge amount of disparate data. In that regard, you have to build models and construct them based on what you know and what you're doing. And it's all nonlinear, it's all it's all homeostatic, and there's these control mechanisms. And yeah, I think I don't know of many. I'm obviously not an expert in this area, but this strikes me as very novel and really important as far as that's concerned. And I do think it will have. Our doc is always looking for these inroads into these measures and what they mean, and we to go beyond just simply, like you're saying, measures that would be biomarkers. You really want to get a little bit more leverage, and this seems.
Well, the irony is that our field has been talking about this all along. We've all along been talking about circuits and talking about processing. So yeah. I think at this point, what we're really trying to do is to transition it away from being a metaphor to being something that is really mechanistic.
Yeah, that's a great point. I mean, exactly. And to bring that, another example is that, a lot of times, right, if you look at any book about brain function, you have these arrows connecting these regions of the brain, and they say, oh, this influences this. And now, instead of just having that sort of metaphorical, you actually have real connections, and there's real communication. And what does that imply? So that's awesome.
And beyond connections, again, really thinking sort of like, what are the operations that are occurring at each point?
Yes.
So
yes, that's, which is fundamentally, each node is not just a node, it's doing something special. And
yeah, exactly. That node is itself a circuit. Yeah. It's doing something. And so we'd like to kind of understand at the bottom layer what it's doing.
Yes. Yeah. And I think this is going to make rapid progress in the future. Yeah, no, this is all very exciting. And
I'm very excited. I mean, so for me, what was really fun about this is that most of us, when we develop in-house software for ourselves, we're all kind of amateurs, but we do it for ourselves and it sort of works. But we decided, that we were really going to make this be sort of commercial grade. And as a result, we're working with professional software developers and thinking through all of the elements of how do you design a GUI that makes sense. And so I had two comments I gave talks in the last few months where I heard two comments, different places, but they were really, really helpful. One of them was by an electrophysiologist, works on animal research, and mitochondria, very much in the space that I'm interested in, but she was very, very intimidated by the whole idea of computational modeling. And she said, I'm really excited by what you're doing, but I need to let you know, I will never use it because the whole thing sounds so incredibly scary. I wouldn't even know where to begin. Okay, so that's comment number one. And then comment number two was from a computational neuroscientist who told me that most computational neuroscientists would rather use someone else's toothbrush than use someone else's code. And that was pretty vivid, I must say. And so, you have to optimize over both parameters. So when you're trying to create a tool like this, it has to be, the usability needs to be intuitive enough that someone who's a really, brilliant neuroscientist, but doesn't know anything necessarily about programming or, control circuits or whatnot, can walk in and, use the intuitions that they know, that they have experimental data to support, and then create these models. By the way, one of the features of the software is that it will export the equations. So when you draw the circuits, it actually generates equations that then are exported. So that's very helpful for those who don't think in terms of equations. Yeah. But for the computational neuroscientists, it also needs to be something that is sophisticated and rigorous enough that they feel like this adds value to what they already do. I think the greatest added value is the ability to directly compare competing models.
Yeah. And yeah, I think that, right, you've hit it on the head in terms of how usable this is. It sort of speaks to maybe the field in some regard. I mean, so you could, just thinking analogously to like, processing packages for fMRI. I mean, everyone agrees on, what smoothing is or this type of thing, but it seems like in this domain, it seems that there maybe people feel like it's still somewhat, even though you have equations and you have relationships, it's somehow idiosyncratic how they implement those in some regard. I mean, it seems like there's still some extra disagreement as to what the standards are, or what the appropriate.
Well, I mean, we're starting with the CBGTC and prefrontal limbic ventral stream circuits because we think those are the ones where you get the most bang for your buck in terms of psychiatric applications. But for each one of these, it's really painful because when you go and look at all the different models that have been worked on and have been empirically supported, generally they are tested within a very specific context. Yeah. And so being able to extract the elements from each one of those and then say, okay, here's the sort of the consensus circuit. It's made clear to me how much this is needed because how can the field progress if in fact, even for someone who's comfortable looking at computational models, it becomes so painful to try to figure out what they all have in common.
Yeah. And it really does. I mean, and not only that, I mean, not only does it give information as to, maybe more refined information as to the specific interactions, but to me, it actually even sort of helps form these general ideas of, the concepts of homeostasis, that it's dysregulated, like, you can describe every single psychiatric disorder as some form of, either becoming too ordered and not responding to the environment enough, or becoming overreactive to the environment in some way, either social or actual. And it seems like, I'm sure this has been done very, very well before, but sort of looking at it that way, this sort of model actually makes it concrete in terms of circuits.
Right. And you can quantify the response to perturbation, first of all. So what is the kind of the degree to which the circuit kind of responds to perturbation? But then also, the control error. So the control error might be homeostatic, but it might also be in the context of reinforcement learning in terms of kind of converging on learning. So it's not always homeostatic. I should mention, it's interesting because when you look at the field of, let's say, computational psychiatry, which seems to have really blossomed within the last few years, it's interesting that when people talk about computational psychiatry, usually they mean either these massive machine learning approaches that I described, which are essentially model-free, or there are algorithms, reinforcement learning algorithms that are kind of more like cognitive science. They are algorithms that predict behavior based on different sort of processing steps, but those are not biomimetic at all. So what you want to do is to kind of marry the algorithms that we think are describing how people are behaving and mapping them onto some actual circuit processing steps.
Yeah. Okay. Yeah, it's exciting. So we're starting to run low on time, but just to finish up a little bit, we've already, I think that, I was going to ask you if there's any big projects or things that are exciting to you. The neuroblox is obviously a big aspect of this, and you certainly are, you're using fMRI, EEG, optical imaging as well, I imagine, or maybe just something.
Well, so for the neuroblox, I mean, basically, we're using a combination of fMRI, MEG, and local field potentials. So, so trying to validate models, in non-human primates, this is where Earl Miller's contribution is invaluable. When you create models, you need to test them, right? So the idea is that it's great, again, to use fMRI as an exploratory measure in terms of accessing all elements of the circuit, but at some point then you need to test that the dynamics you think are occurring are actually occurring, and that's where the non-human primate experiments come in.
Okay. All right, well, so maybe the last thing, is there any sort of advice that to young scientists? I think you would be a great example for young scientists who sort of, scientists are, it's all about forging your path. And is there any advice that you would give to young scientists going into brain mapping, for instance, or even neuroscience in general?
I think that there's a tendency I've noticed in students is that there's a general tendency to follow established pathways. The thought is that if something is hot right now, that's what you ought to be doing because it's hot. And my own thought is that generally it's more fulfilling and interesting to think about a different, not what's hot, but what's important. And the other piece of advice I would give is that in following your decision for what's important, it's wise not to get too caught up in artificial distinctions of fields, because the best tools might come from many unexpected places. As I mentioned, many decades worth of modeling physiological control circuits completely out of the brain were very helpful in thinking through what might be happening in the brain. Population dynamics, thinking about predator-prey type models turn out also to be useful in thinking about regulation and so forth. So thinking across scales, being able to think across modalities, different fields, I think generally having a kind of a flexible approach and being kind of more question-driven as opposed to technique-driven, I think has been helpful.
Yeah, that's great advice. And right, if people are technique-driven, they become married to a technique and their questions are very limited. And in fact, we're still, the big question, right, is the big problem is coming up with the best questions. And I would say that the corollary to that is also allow yourself to be surprised, because I also think a danger is getting very invested in hypotheses and not noticing that often some of our greatest discoveries happen when the data surprise us. And being kind of, again, sort of cognitively flexible enough to embrace that rather than suppressing it is a good idea, I think. Yeah, and that's a skill, I think, also that comes over time. And I totally agree with you. That's great advice. Yeah, all right. Well, thank you very much. Thank you so much, Peter. Nice to see you. Good to see you. Next time in person, I hope. Yes, definitely, definitely.
Neurosalience is brought to you by the Organization for Human Brain Mapping. This week's episode was produced by Anastasia Brovkin and Alfie Wine.